Yeah, so as I was saying, I'm not a stranger to Christ. I mean, Christ University is deemed to be. Say stranger, no stranger to Christ. Okay, that may be a. Uh, so the thing is, yeah, I've been giving. I started actually in 1990s. That's probably 30 years ago, giving refresher courses here in the physics department, and so there were many students from and teachers from other colleges. So started like and on and off. I've been giving many, including in astrophysics and other topics. In fact, some of the people were recalling that about some of my lectures about 10 years back, and the year of physics 2005 also, had given some lectures here. So actually, I've been quite involved, and more recently in 2016, I had given a course in generator in cosmology to MSc and BSc students. That was for a certificate course, and also on and off giving courses and lectures. And association goes back to quite some time. And also, as Arun mentioned, I guided him, and also now some other students are also there from Christ for their PhD. So in fact, teaching is also one of the. It's not always research. Research and teaching should go together. I've always felt that way. So you might have heard some 300 papers, and all. It doesn't mean we should not teach. In fact, I've been quite active in teaching, if I may say so. Rocket dynamics course I've been conducting in St. Joseph for 15 years. The rocket dynamics interdepartmental course sponsored by ISRO, and a number of other things. So yeah, so that's what. And again, some remarks about the Nobel Prize, this thing. And in fact, we in our newsletter, every time it's mentioned all the Nobel Prizes for the past so many years. And actually, the tradition started even earlier. When I was a student in IIC, student means I just finished my PhD, and it so happened in the year 1978. The Nobel Prize was in physics, was shared by Kapitsa. Kapitsa, I think you all know, liquid helium, liquefied helium. So he was a Russian origin, and he shared the Nobel Prize with Penzias and Wilson, two Americans from Bell Labs, and their work was on the microwave background. Okay, uh, cosmology, cosmic microwave background. And why they were clubbed with Kapitsa? Because they also use low temperature physics antenna, and things like that. And the discovery of the microwave background was a big discovery. Okay, and because for the first time we are seeing the radiation from the beginning of the universe, the Big Bang, and it was there all over. That's what. And these people who got Penzias and Wilson, they were not scientists. Okay, and actually they were engineers. Uh, they were telecommunication engineers hired by Bell Laboratories to reduce the noise because that time satellites were already there. Okay, and so to improve communication with satellites. Now also we require satellites. In fact, mobiles means if everybody uses mobiles, 5G and all, we need satellites because the tower has a range, finite range. That's why Mr. Musk is launching so many Starlink satellites. Okay. So that's the reason we need thousands of satellites. Okay. So that's why to improve communication. You want remote places like Tonga and all, get to earthquake. That's the only way of communicating. So all this, so this Penzias and Wilson, got, and so in IAC, the physics department, they knew about Kapitsa's work, but then nobody had known anything about what is this microwave background, Penzias and Wilson. Huh? And it was a purely accidental discovery. And when they got Nobel Prize, they didn't know, the importance of what they had discovered. It was told to them, it's a very important discovery. Okay, it was one of those serendipitous discoveries. Okay, which means purely accidental. So in fact, I was the nearest to that subject because having worked in my work had something to do with the early universe and all that. So I was asked to give that part of the talk. So it began like that, and then it started. So similarly in 1980 also, uh, Fitch and Cronin got Nobel Prize, for their work on CP violation, and that has an important consequence for cosmology, in the sense that we like to know, it's still an unsolved problem. We like to know why there is more matter than antimatter. Hardly any antimatter in the universe. Suppose there are equal amounts, we won't be here because everything would annihilate. as you know, as students of physics know. So what is the reason for that? And the work of Fitch and Cronin gave some indication that some symmetry called CP is violated. This gives the possibility of having an excess of baryons over the antiparticles, antibaryons. So 1980, had given that, and then after that I shifted to IA, and 83 Chandrasekhar got Nobel Prize. Yes, Chandrasekhar. So many people wanted to know why he got. You might, instead of astrophysics, they wanted to know why he got, because they wanted to know how this white dwarf mass limit comes. Okay, and in fact, our director at that time, Professor Bhattacharya, he told me, "Oh, you have to explain it in quite detail because many of us have no idea about these things." Okay, so anyway, that was also, then it became a regular feature after that. And of course, many times particle physics also 88 prize was given for this thing for muon Newton. No, 85 prize was given to von Klitzing for the discovery of the quantum Hall effect. So I started writing articles also on that year's Nobel Prize, and it came in many newspapers also. This part of the outreach. Now there's a lot of talk of outreach. You know, we were doing outreach even without being formally, in fact, when the Chernobyl disaster took place, I wrote a series of articles on nuclear because we are thinking of importing those kind of reactors, for our purpose, gigawatt reactors. So anyway, so yeah, so there have been, it became a regular feature, in every year giving this kind of thing, and sometimes even chemistry. I used to give talks on the chemistry thing. In fact, one example was in 1996 when the prize was given in chemistry. That time, the prize in physics was given for the Richard Richardson. You might have heard, for the liquefaction of helium-3. Okay, helium-3 is difficult to liquefy to become a superfluid because they need much lower temperatures, and they managed to do it. And so, that was the physics prize. And chemistry prize was given to for the discovery of buckyballs, Buckminster fullerene, a new form of carbon, new allotrope of carbon. You have graphite and diamond, and this familiar, but there's also graphene. There's also graphene, and that was a big discovery. The new state of this buckyballs, Buckminster fullerene was the new. Graphene came later. In fact, I covered that in 2010. People wanted to know what is that kind of thing, and so Buckminster fullerene was important. Sir Harry Kroto, got Nobel Prize along with Richard Smalley from the US, okay, and Robert Curl, and that had some importance in astrophysics because the motivation for that came from astrophysics, because there are stars called red giants, and they shed a lot of dust, carbon, the form of carbon, and they got helium, excess of helium. Okay, they eject, and so it was thought that some this carbon might condense in that kind of environment, in that kind of temperature. So these people thought if they reproduce those conditions in the lab, they may get some new form of new type of substance, and that's what they found. And later on, it was identified in astrophysical thing, the so-called diffuse interstellar band. There's a absorption feature in the ultraviolet 2218 angstrom that is attributed to Buckminster C60, okay, and C60 plus also. Imagine 60, carbon atoms, in one of the things. Now, of course, they're going to C120 and C240, and buckyballs have played an important role also in material science, as you know, in getting better superconductors and things like that. Many properties, okay, the tubes and things like that, nanotubes and things like that. So there have been so many cases where so let's come to this year's Nobel Prize. So I just gave a background because there's something, it's become an annual feature to give talks on many of these things. And I remember when Chandrasekhar got in 83, the medicine prize was given to Barbara McClintock. He was himself quite old when he got Chandrasekhar. He had to wait 50 years after his white dwarf thing, half a century. And Barbara McClintock was actually also older. So she got for the so-called jumping genes. I don't know if biology people are here. You can have, gene, this thing, flipping, from one this thing to another state. So I gave something on that also. Okay. And so it's become a more or less. So the thing is, one should have a broad interest in these kind of things. And in fact, I was reading recently, there was, this has to do with Christ. somebody from the chemistry department, I think it came 2-3 days back, that found normally we all cry over split milk or spoiled milk, but here, they're trying to see uh, uh, how to prevent milk from getting spoiled, or how to see whether it's getting spoiled by measuring the so-called pH. Okay. The pH of milk is supposed to be, it's of course alkaline. If it turns acidic, it will get spoiled. That was 6.5. It's supposed pH is supposed to be between 6 to 6.5, and so so they had a technique to measure it using uh, using colored materials which change their color, halochromic materials, if I remember right. If they change their color, with the pH, and using these plant pigments. Okay, anthocyan is what they called. Okay, and so with that, hibiscus flowers and things like that, and coating it on a paper, seeing whether the pH exceeds some value, it will become decreases to some value, the milk will get spoiled. So how to prevent that? So in fact, billions of liters of milk are wasted every year because of getting spoiled. And as you know, India is the biggest producer of milk now, thanks to Dr. Kurian, the early work of Dr. Kurian at the Gujarat Cooperative Society. Okay. So, so this I mentioned in passing. I don't know, are you in the audience, anybody in the chemistry department mentioning their work? It just came, no, okay. So I just happened to read this come from Christ, and this is some practical thing. You see, this is some practical thing. Normally, much of the research, the criticism is all exotic and all that, but here there's something of of practical application. So I thought I should mention that, and since this is an audience in Christ, I thought I should mention it. Okay. So anyway, it doesn't matter. I don't remember their names. Chitra and Vinod. Am I right? Huh? I'm right, is it? Okay. So anyway, this is just a recent kind of thing. Okay, I won't mention some other thing. Let's go to this year's thing. We'll come back to the other things later. Okay. So the order in which we follow here is physics, chemistry, and medicine. Although the usually announcement of the Nobel Committee is medicine first. Medicine is always announced first, then physics, then chemistry. But here I'm following PCM because PCM is our Indian, including. So instead of physics, chemistry, maths is now physics, chemistry, medicine. You're following in that order. Okay. All right. Although I do wish, I think maybe it's a good idea to have PCM with medicine. It may improve the employability prospects at least. Many. So because many applications in medicine now are involve physics. Okay. Especially for heart and breathing and so many things. They involve physical principles. Okay. So it went to three people. Uh, you can see, it is Alan Aspect, the French guy, and Clauser, John Clauser, and Anton Zeilinger. Okay. And Aspect is a French man, Alan Aspect, and it was this. It's a bit nearer. Can I come? Okay. You can place it a bit nearer. Okay. Yeah, it's okay. Can make it a bit. Okay. Okay, Alan Aspect, actually the work was done in 1981. The paper by Alan Aspect, Grangier and others. And so actually, it involved so-called, it was a very basic experiment, okay? And it settled a long held kind of argument about quantum mechanics, okay? So-called entanglement. That is, uh, what I think physics students of BSc also will have known about quantum entanglement of photons. So this is some peculiar phenomenon in quantum physics that, suppose a particle, two subatomic particles, okay, it could be photons. Suppose they have they have interacted, okay, they had some interaction, and say this, then they separate, okay. it turns out the measurement of, if you measure, an observer measures a particular property, spin or polarization of one particle, is instantly it fixes the thing, correlates with the other particle, however far apart they are. This is called entanglement. Literally, they get entangled. You know what entanglement means, no? Okay. And what is interesting is it is independent of the distance, however far apart they are separated. And actually, Schrodinger drew attention to this, okay, somewhere the famous Erwin Schrodinger in around 1935. And so then Einstein felt that it is not, it looks funny. How can it instantly affect the other particles? So he called it spooky action at a distance. They wrote a paper, Einstein, Podolsky, and Rosen, two of his assistants that time. The three of them wrote a paper together, and they said that this looks rather odd. Although they considered momentum and position as two kind of things. How the measurement of these two parameters can fix the thing on the other particle, fix the correlate. Yeah. So that's what, okay. So that's, okay. So groundbreaking experiments with entangled quantum states. So these are called entangled quantum states because, they have a state, as a property, different properties, spin, polarization, other points, left or right, up and down. So many possibilities are there, right? Okay. So that is the thing. So with the entangled states, where two subatomic particles they behave as like a single entity. Okay. Although they are widely separated now, even when they are separated over large distances. So their properties are correlated, however large their separation. So although Schrodinger drew attention to this, a similar 1935 paper of Einstein, Podolsky, and Rosen, EPR is called, seemed to be dismissed it as spooky action at a distance. So it looked incredible that a measurement of, say, a spin state or a polarization of one particle would instantly imply on measurement the same correlation in a distant particle, okay, once they are separated. So, of course, Niels Bohr, of course, supported, as you know, the argument Bohr and Einstein. Bohr preferred the usual quantum mechanics. Again, the measurement problem, the collapse of the wave function and all that. Einstein thought there's something, quantum mechanics is not complete if you have to go to this kind of probabilities and things like that. So he thought there must be some underlying determinism for all these things. So that's why so he said, and as you know, Einstein started special relativity. Signals can't exceed the speed of light. So how can measurement of one particle instantly affect the corresponding measurement of a property of another particle? How can it instantly affect? That's why he called it spooky action at distance. Then some other people they try to find alternative methods to explain this. Like among them David Bohm and others. So of course I'm not going to go into a detailed kind of thing because there are a lot of things which people are still saying about these things. So we are not, even the experts sometimes differ on some of these things. So the thing is about aspect is sometime around 1965, John Bell, a physicist working at CERN, he tried to settle the kind of these things. So he said we'll come to that also. That he said that suppose something is classical, there should be some deterministic kind of thing in the correlations. So if it doesn't follow that, then it in the conventional quantum mechanics, he postulated that there should be some some statistical this thing. There's a correlation between these two particles. That is, if you measure here, say the spin, say left or right or up and down, the corresponding thing should appear in the other particle also much further away. Okay. So that's what he tried to say. But then he showed that quantum mechanics, the usual conventional quantum mechanics, predicts a very distinct kind of correlation, okay, which is not there in the classical or in the other alternative pictures, okay. So for a long time, people were the experiment could not be done. So the experiment was not feasible or not thought of. So the importance of Allen aspect is he translated all these things into reality. Okay, that is the important thing. He translated into reality. What people were just guessing, and making suggestions and estimates like John Bell. inequalities. He actually was able to do the experiment and show that it correlates, as predicted by quantum mechanics. So Aspect first translated theory into experiment in 1981 by verifying that entanglement of two photons moving away from each other. They are moving away from the source, okay, two photons moving away from the source. And when they were separated, say, over 12 meters, he undertook the measurement of the polarization, okay, of these two photons. And then he showed that the thing is they went in opposite directions, right? And that they covered sufficiently, they're sufficiently far apart. And the measurements were quite were done with moving of a thing, that is, they are moving away from each other, and the photon polarization correlation agreed with that of the conventional quantum theory rather than of the alternative ideas. So this was a blow to the Einstein-Podolsky-Rosen thing, that this action at distance takes place. That is, however far apart they are, their properties are correlated. Okay. So there's something quite thing. So the so-called Bell's inequalities were found to be consistent with the usual quantum picture, not with the usual. Of course, here, I mean, I don't want to go into the details. One example given by Bell was the so-called Bertelsmann socks. So this, I don't know how many of you have heard of that. So Butterman socks is, of course, you all heard of the Schrodinger cat. There are so many things in quantum theory which are, you know, examples have been given. Okay, the cat being both alive and dead and things like that. Okay, so the thing is, unless you make the measurement, you can't be sure which is the thing. Otherwise, the superposed state of both dead and alive. Okay, so Butterman socks is like this example was given by Bell. So Butterman was a friend of or a colleague of Bell. He was noted for wearing socks of different color in each of his feet. So say he wore red in one, say on the left foot, and green on the right foot. Okay. So the thing is, since you know that he is, he wears socks of different colors. Okay. So suppose now somebody looks at his left, this thing, left leg, okay, left ankle, you see it green, then you can be quite sure that the other will be red, right? You can be quite sure the other will be red. So you already have the kind of thing. This is what you will expect classically. Okay, this is what you will expect classically. Butterman socks. Okay, it's an example of. So you have the information already that that he wears socks of different colors. So if you make observation on one foot, you know the other foot is going to be the other color, another color, right? So you know it already. But here it's not like that. It's not classical like that. Okay. So here, classically, what you expect is, quite different from what you see, the correlation you see, which is actually measured. So in brief, this is an example of Alan Aspect's thing. Okay. Butterman socks. Okay. The so-called Bell's inequalities were found to be consistent with the usual quantum picture. This supported the spooky action at a distance, which correlated the measurements of the particles moving away from each other. Okay. Can you, as an example, you can measure the spin up or down, left, right. So you see the different possibilities are there. So quantum mechanics gives a unique, prediction for the correlations between the two. Okay, this, yeah. So this was done in 81, very long back. So you see, it's a very belated Nobel Prize going after. And also Anton Zillinger, who shared this year's prize, and along with, he's from Vienna. Alan Aspect is a Frenchman. This is from Vienna, along with Greenberger. So Zillinger, Greenberger, and Horn, they did like, of course. See, what happens in many of these Nobel prizes is many of the co-authors are not leaders of the teams get. It also happened in the Buckminster Fullerene, even the Higgs particle. Maybe there are 200 authors of the paper, right? But only the leaders of the team get. Okay, so that's what happens. So you have Greenberger and Horn not being mentioned. So Anton Zillinger get. What they did was they carried out the entanglement experiment with 3 photons. Okay, not with 2. Then the correlations are, the probabilities and correlations are more complex. But they showed that that also it is satisfied. There also the quantum predictions of quantum mechanics are satisfied. That's what they showed, and they have extended to more number of particles also. So that was and quite recently, a Chinese satellite experiment. So how far apart should they separate to see that their properties are correlated? Okay. Will it be also valid for very large infinite distances? Sir, unless you make the actual measurement, you can't be sure, right? So some people made that question also. So the Chinese satellite experiment, it verified the quantum entanglement over a distance of a few hundred kilometers. Okay. Because satellites, are a few hundred kilometers above in orbiting. So it was verified that this correlation occurs there also, okay, as predicted by quantum mechanics. That is, even after the particles break away, the the characteristic properties of the measurement parameters like the spins and polarization remain connected, correlated, however far they are separated, as if by some invisible piece of string. Okay, something is happening to make them correlate with each other. Okay. So this counters the Einstein's view. Although of course even aspect said that Einstein should be given credit for raising the question in the first place. Okay, he raised the question and people had to settle it. So you must give credit to the Einstein Pollar-Rosen paper for raising the question. Okay. Yeah, so this is an example of Anton Zeilinger. He concluded more tests of the inequalities. He created the entanglement pairs of photons from shining a laser, a special crystal was there. And I won't go into the details because there are, and random numbers of this thing to shift between. They have to be sure, that it's quite, it is independent of many things. So they try to make uh, they correlated with different numbers and things like that to see. Okay, so signals even from distance, you can even get uh, there are even other examples. They in fact involved even celestial bodies, photons from that to see that they're still correlated. Okay, so anyway, I won't go into that. And there's a lot of things, and people are still discussing some of these things, whether it is the significance of these. And I should also say that this has got implications. Not only is not apart from the thing as a pure, uh, 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 from pure science point of view, pure physics kind of thing. There's a lot of applications, these kind of things, because see, when you create an entangled a system of entangled states, it sort of helps in things like quantum computation. Okay, because you're prepared a set of entangled states in correlated with each other. So parallel computing becomes, as you know, uh, apart from the usual, in the usual computing you have bits 0 and 1. Here you have few bits. Okay, so it can be either 0 or 1. So more possibilities are there. So now if you prepare these correlated systems, okay, it may be atoms aligned with the magnetic field or things like that. You create such a kind of thing. You can do parallel computing. Okay, you can do parallel computing because there are so many possibilities now. Okay, you can solve many, equations and say fast, quite fast, because of course there are again some constraints there. I won't go into the details. For example, it has been known for a long time that right from the days of the when quantum measurement was discussed, that the apparatus should be classical. See, apparatus can't be very small. The apparatus must be substantially larger than the quantum thing being observed. For example, suppose you got say if you do a double slit experiment. Suppose the screen is very light. Okay, it will start shaking, right, vibrating when you make the measurement. So the pattern will be disturbed. So there's a criteria for how much should be what should be the size of the screen and things like that. Okay, so that's another strange aspect in quantum theory is the measurement. The apparatus is classical. We assume it is classical, but the system we are examining is quantum. Okay, this always kind of leads to a, so you got an entanglement of a classical state, the apparatus and the quantum state. So this again leads to some, I won't go into the thing, but there are lots of interesting things here. So in fact, especially in quantum computing, these things have been applied. Suppose you want to do the operation over a time t, it turns out there's some minimum energy required for that kind of thing. Okay, in terms of the Planck's constant and the time scale of the operation. Okay, so anyway, there's some interesting things are there. Anyway, I won't go into that. So it has led to a lot of spin-offs, including quantum cryptography and things like that, quantum security, things like that. So because they are correlated, you can prevent any any change in the thing, can be easily detected. So the thing is, if somebody is eavesdropping or something like that, or somebody is trying to interfere with the system, it will be revealed. Okay, because the correlation will be spoiled, right? So then you can. So this has led to what is called quantum security and things like that. So lots of applications are there in connection with security and information security and many similar things. Okay, and telemetry also in telecommunications and also and other things, the sensitivity of the thing and the precision in the telecommunication. So there are lots of spin-offs here. That's why Aspect has called it the second quantum revolution. Okay, the second quantum revolution is called it because it led to so many new things. So I should also mention these things that there are other applications of these things in quantum computing, quantum cryptography, quantum security, so many things. Okay. Cryptography is nothing, you send secret messages. So here again, you can imagine how with some correlations you can create a particular, particular message or a particular thing. It's very difficult to for an outside person to decipher what you have done because your system is very unique, correlated in a very particular way. So all these applications are there. Okay, all right. So maybe I've concentrated too much on physics, but let's go to the other things also, which are quite important. So we'll see the chemistry Nobel Prizes. Caroline Bertocci, she is a lady, and she is the eighth woman to get the Nobel Chemistry Prize. Okay, it starts with Madame Curie, who got in 1911 for the chemistry prize for identifying polonium. She shared the earlier one in physics with Pierre Curie and Henry Becquerel for the discovery of radioactivity in 1903. And 1911, Marie Curie got the first woman to get chemistry prize. So she got two Nobel prizes actually, and her daughter followed Irene Curie in 1905. You can try to see who are the other. Okay, and Dorothy Hodgkin is a famous crystallographer who unveiled the structure of insulin and so many complex molecules. She had many students from IISc who later rose to high positions in IISc, including Professor Vijayan, who became the head of the molecular biophysics unit, Ramaseshan, was the director of IISc. She had several distinguished students from IISc and Indian students, right? So Dorothy Hodgkin. So Caroline Bertocci is there, and Morton Medal and Barry Sharpless. This is the second time Barry Sharpless is getting the Nobel Prize. He got the first one in 2001 for the synthesis of uh, chiral synthesis of hydrogenated molecules that time. He shared with two others, Japanese one, Niori, and Novels, American, John Novels, for their work on these are catalytic, this is chiral catalysis. That means you created handed molecules. Okay, molecules either left hand, they change the polarization of right, either left or the right way. So they were able to synthesize many of these chiral molecules. They had some agents for that chiral catalysis. And so the Barry Sharpless in 2001, remember I gave a talk in ISC on this Barry Sharpless work because it was of special interest because thalidomide was one of the, you might have heard of the thalidomide catastrophe. They produced the molecule of the wrong-handedness. Okay, so it affected many women, with it was to reduce pain and all that. So they miscarried and created a lot of the thalidomide controversy. So again, handed molecules, are important. Molecules are one. They're identical in every way except that one is left-handed or the other is right-handed. They change the polarization the left way or the right way. In fact, sugar, for example, your sugar is dextrorotatory. It changes the polarization in a right-handed way, sugar. Whereas amino acids in at least in living systems are all left-handed. Amino acids are left-handed, like most of the amino acids, guanine and all those things. Yeah, they are all left-handed. So that's a mystery why they are left-handed and sugars are right-handed again. Okay. So work on linking molecules together, which became known as click chemistry. Click chemistry, what do you join simpler molecules to make more complex molecules in an instantaneous way. Okay, that's what they find. That's why it's called click. So work has been, so that is, we must know the properties quite, there's something called bioorthogonal reactions and things like that. So the work has been well applied in tracing various biological processes while exploring body cells. So they created the possibility of creating these compounds inside the cell itself. Okay, and see that the drugs go to the right place and things like that. So it has seminal applications, for instance, in creating cancer treatment drugs. So click chemistry involves linking molecules in our cells. Constantly this is happening. Okay, because the DNA is the master molecule, the blueprint. So we have the transfer RNA and the messenger RNA. So the messenger RNA brings the thing to the site. Okay, so the proteins are created on the site. Okay, for example, your liver, for example, if we eat so many things, the liver has to digest all that, right? So if you want to build a chemical factory which does all that the liver does, it should be the size of a city. Okay, I'm just telling you. So it all does it in 1 liter volume. Okay. So and not only that, once they're all catalysts because you have to digest the food. You eat pizza, you eat vada, you drink this thing. See, how does your body take care of all that? Convert it into, say, your glycogen, which you can use for your energy, producing energy. Okay. So that's quite a complex process. So these catalysts are involved, And how are they're made at the site? Because the RNA, the DNA is the master molecule which has the information. So the transfer RNA transfers it to the messenger RNA. Messenger RNA brings it to the site. And the amino acids, there are 20 basic amino acids. And as you know, and from that you can create so many proteins. You can work out how many combinations are possible. And then these proteins are produced and they have to be destroyed. Otherwise, they'll keep accumulating, right, and poisoning your system. That again, there's a catalytic process. So these things are made inside the cell. And what these people have done, you can artificially, you can mimic that, with the simpler molecules and make them produce them as on site, the complex molecules. So Barry Sharpless and Morten Medal from Denmark, they worked independently but together laid the foundation for click chemistry. It's an important biological discovery tool that allows us to see and develop new molecules that we are not, sometimes we don't even know such molecules exist. Okay. Same thing happens in our body. We don't know the structure of all our molecules, the ribosomes and all that, how they produce all these things. Okay, this is some example. I won't, I'll skip that. Okay. So we are already come to medicine. Okay. So this is called click chemistry and okay. And Barry Sharpless is getting for the second time. Okay, 2001 now he's getting in 2000. Again for synthesis of compounds, click chemistry. That time he did for clicking the chiral molecules, that is 100 molecules. Now it is more general. Okay. All right. So 2022 Nobel Prize for Physiology and Medicine. Award for discoveries concerning genomes of extinct humans. And of course, you all heard of the genome revolution, right? Now you can read the blueprint genomes, okay, of any individual or any organism. We have got something like 4 billion, in your uh, in your gene. Now you've got nucleotides, okay. Nucleotides are, the four basic ACGT or the DNA molecule. So your typical this thing gene has something like 4 billion of those nucleotides, okay, and they are responsible for making all your proteins, all your things, that's why your fingerprints are distinct, because it's unique for everybody. And so you can read the genome, the positions of all uh, in the gene, the positions of all these nucleotides. They are in groups of three. Okay, that is called a codon. So it can be CAG or ACT. A and T normally coupled together. C and G, okay, also coupled together. These are the four nucleotides. Okay, so that's what happens. And then so you're able to. So we got genetic defects, say for example, and say for example, you have got diabetes. The congenital diabetes. That means you're unable to make insulin. Okay, insulin is made in the pancreas, so-called islets of Langerhans. Islands. So in fact, sometimes in geography quiz, we ask where are the islets or Langerhans. They try to say, oh, is it in near West Indies or is it here? It's in your body, right? Islets or Langerhans. Similarly, Havesian canal is in your body, right? Important. Schlem canal is in your eyes. Okay. So there are many such geographical names in the thing. So islets or Langerhans make the create insulin again from your blueprint. But you know this in some there's a genetic defect in some individuals. Okay, that is hereditary. That this particular thing is defective, so it doesn't make insulin. So that's why your problem with sugar. Okay, and of course there are problems with other sugar kind of thalassemia is there, for example. Okay, and some people are not able to digest lactose. Okay, that also has got so then the idea in sort of reading the genome is to see if there are what defects are there and then try to rectify it so that you don't have to take insulin, can be corrected, genetically corrected. So the Human Genome Project, led to a lot of revolution. They're able to read the genome, okay, and for every individual and say what is your, what are the things you may be exposed to, what are the your weakness kinds of things. But is it audible what I'm saying? No, huh? It is okay, right? Because I can't shout loudly. I'm not used to the. That's for political. I can't shout loudly. So, so yeah. Okay, so what I was telling you, yeah, so the Human Genome Project, has unveiled the DNA can be sort of examined, your DNA, and you can tell what are the things. And of course, now it's been applied to so many things. Even forensics has been applied. Okay, recently I was not only to your not only to your genetic history of your what are your congenital defects or what are the things diseases you may get, okay, and things like that. Uh, but even say forensic medicine. Recently, I was reading some person, somebody was incarcerated. That's the word used in the U.S. It has to be a colored person, right? So 36 years he was incarcerated, accused of some murder. Then some DNA, test showed that he is not the culprit. so, so with DNA, forensic DNA, you are able to tell, who could be some trace of it, maybe a hair or something at the site. You can tell this is the person who has done the thing. So at last, this fellow was released after 36 years. Can you imagine? Huh? So he got justice. He was lucky he was not sent, put to. In fact, in fact, I don't know. We think some of these countries are very civilized, but there are people on death row for 20 years, 30 years. Okay, and at least they are executed after that. 25 years, 30 years. They are given lethal injections. We are better that way. We release the Indra, that Rajiv Gandhi is right after 30 years. But in the US, they are still put to after being incarcerated for 30 years. So forensic medicine, it has been applied to the DNA thing, and but then all these DNA now samples are freshly taken; they are pristine. But what about DNA of some say fossils, for example, or of ancient? There have been we had our human history is quite complex because we don't know who are the origin of man is still we are very sure of the origin of the universe. What happened right from the picosecond we know, right? Now we think say 1 second after the universe began expanding, the temperature was so much. They're able to now we are very sure, right? Hmm? But we are not sure about the origins, how man originated. Where exactly is the dividing thing from, say, the primates to the Homo sapiens? Where exactly is that? They keep pushing it back, maybe 100,000 years earlier, and each country claims they are the, people having the thing. Sinopithecus is the China man. Okay. Yeah, even our Indian thing, Ramapithecus Punjabicus. One of 14 million years ago, one primate was there. Okay. Java man is there, China man is there, Australopithecus is there, Ginjanthropodus is there in Africa. Okay. That is now, they believe that, see, anything primitive, they'll shift to Africa. They'll say Africa. including any AIDS and all. Africa, it is the thing. Okay, so anything started in Africa, right? And then, so the earliest man started in Africa and then migrated outwards. But we are still not sure of the thing. So only we should have some method of identifying, of also, just as the Human Genome Project, analyzes the recent DNA, right? The pristine DNA, fresh DNA. Can we also analyze the traces of ancient DNA? Okay, so that's what this year's physiology medicine Nobel Prize is about. Okay, so he's the founder of the new discipline. Of course, he founded it 20 years, more than 20, 30 years back. He's dubbed paleogenomics. Genomics is the unraveling of genes, genomics, okay, study of the DNA. So paleo means ancient, Paleontology comes from that, fossils. Paleo means ancient in Greek. So paleogenomics, okay. So paleogenomics, which maps the genomic sequences of ancient DNA samples, okay. Talking of this, apparently there is a new institute in, I think, Madurai Kamaraj University, I was reading. Okay. Institute for Ancient DNA, analyzing ancient DNA. It's a new institute for ancient, analyzing ancient DNA. Okay, it's an oxymoron, new and ancient. So here what they do is they had some excavations, okay, of in Tamil Nadu of some, you know, uh, animals, carcasses, fossils of humans also, and they're analyzing it to see what was the history of this, okay, excavations. And they're collaborating, it's Madurai Kamaraj University, I was reading. They're collaborating with Harvard, Adam Rice, who's an expert on the ancient genetics. So it's come into the picture that if you want to know how our, what is our ancestry, and things like that, how people mingled, okay, and things like that. Your analysis of your DNA will tell you that. Okay, that's the whole idea. So it's now become it. What was started 25 years ago, it has also come to our country also. We got a institute for ancient DNA, department for ancient DNA. Okay, some Kumaresan is in charge of that, I think. So anyway, so this will come in our thing also. Okay, so so what do we hope to? So we sequence also ancient DNA, and of course, but it's present in trace amounts. See, the fresh DNA, a lot is available, right? A lot is available. I can take a sample from somebody and analyze it and tell the genome sequence. Okay, Sanger has done a lot of work on that. Okay, Sanger Incorporation is a private company. Okay, and so, but then how do you amplify this small amount of DNA? How is it amplified? So I will tell you that also. That again, it goes back to another. Is it okay? Pablo pioneered. Let me finish that. Okay. However, much of this sequencing from DNA of living species from 1990s, the genome sequences have been with fresh, pristine, and uncontaminated. The DNA which has been exposed from the fossils and others has been exposed to a lot of contaminants from other sources, right? So you can't be very sure about the thing. Okay. So what this Pabo, this person who got the Nobel Prize this year, okay, Swante Pabo, whether his father also got Nobel Prize, Carl Bergstrom in 1982. So it's of course his father shared with two others, but he is getting it alone. So it's an improvement in the family, probably. I don't know. Okay. They are Swedish, by the way. They are Swedish, by the way. Okay. And of course, you may say Swedish Academy only awards these Nobel Prizes. So is there some, I don't know, we can talk about that later. So anyway, so Paceway, he pioneered the sequencing DNA of dead or extinct species, okay, involving non-living tissue, okay, first. Only minuscule and, contaminated samples have to be analyzed, okay. And the DNA from, say, mummy, he started with mummified Egyptians. Because Egyptians, preserved their bodies very well, preserved their, you know, dead very well, mummified, And how they did it, that's again a secret. One blue dye they use, only quite recently they analyzed the composition of that. Why it's been permanently blue for thousands of years? They knew some chemistry apparently. They knew some chemistry apparently, which has only now been understood. So some of these things, their motivation might have been different, but they also made some, discoveries or some things like that. Okay. So only minuscule quantities are have to be analyzed, and the DNA is mummified Egyptians. was first sequenced, following later from our ancestral Neanderthals. So you might have heard of Neanderthals. Neanderthal family is one of our ancient they coexisted with humans. Okay, they are from Europe mainly, whereas the humans, are started from Africa and they migrated. Okay, those I don't know how they migrated. No planes, no trains, no metro, nothing. They anyway managed thousands of kilometers. That's another. Okay, so of course a lot of time anyway. So maybe slowly they moved over. Okay, technique. So another important discovery comes here, which was done long back. A technique to amplify what DNA is available, so-called polymerase chain reaction, PCR, polymerase chain reaction. This is a technique developed by in 1993 Nobel Prize was given to Carey Mullis. Okay, for amplifying DNA. Oh, this polymerase chain reaction, even if you start with a small sample, okay, of the whether it's fossil or contaminated, you can amplify it with this chain reaction, polymerase chain reaction. As you know, a chain reaction always multiplies the thing. Even the fission chain reaction, the number of neutrons get amplified. So here you can amplify the DNA with this chain reaction. Okay, so that was an important earlier discovery of Carey Mullis. Okay. And so in 2010, the direct sequence of the genome of a Neanderthal was published by these people. Okay. So to know our ancestry, how much of our DNA comes from Neanderthals? Okay. And Neanderthal, named after a place in Germany, the cave in Germany. Okay. That is how you have the Neanderthal. Similarly, we have the Denisovans. They also coexisted with us, but in Asia. So that is a region in Siberia. Okay. That's how it is the Denisovan. So that's how they get the name. So this technique of analyzing ancient DNA was carried over to analyzing DNA samples of Neanderthals and Denisovans. So that was the paleogenomics is called. So the Neanderthals as a distinct biological subspecies was first identified in 1856, long back, from local bones discovered in a limestone cave in Neanderthal, okay, in a valley in Germany. That's how the name comes. Okay. And the Denisovans is named after a Denisova cave in Siberia. Okay. So they are also another early strain of human forerunners. Okay. And the DNA extracted from just a finger bone, in this case, was sequenced. The DNA extracted from a finger bone, any part of your body, because now you can amplify it, and then you can sequence it and see how they were different from ours. Okay, how they were different from ours. And so these methods and techniques of analyzing primeval DNA of early distinct different humanoid species and establishing their genetic connection with modern with modern humans underlies this year's prize. So now you understood what the prize this year was given for, right? Okay. Hmm? Okay, so this is the thing, Neanderthals, and so see how they are migrated, right? And see the map is also given here. Okay, how they went to different from Siberia, and Europe, there was apparently interbreeding between Neanderthals and Denisovans and humans. Okay, and so it turns out some fraction of the DNA, for example, maybe 5 to 6% comes from Neanderthals, and especially in Southeast Asia and also from the Denisovans, some fraction of the DNA has come, small percentage. But this probably enables us to withstand severe winters because Siberia had severe winters, things like that. So, so there are some spin-offs from this genetic mixing, of the DNA. Okay. So Neanderthals, and they say no, you inherit your DNA from your parents, from your grandparents, etc. Here we are inheriting from a very so species existed 100,000 years ago or 200,000. Isn't that interesting? Huh? So so the Neanderthals and Denisovans originated in Europe and Asia. However, the genetic line leading to humans originated in Africa. They always blame Africa, and this becomes dominant. Okay, and where this line from Africa encountered Neanderthals, there was interbreeding, and some Neanderthal DNA variants were incorporated into the human gene pool. They talk of gene pools. That is your accumulated genetic this thing, what heritage. Okay. So the genetic markers can still be detected. 1 to 6% of this DNA is present in people of Europe and Asia, while in Southeast Asia, 5 to 6% of Denisovan DNA is present. These studies also showed that Neanderthals, uh, who mostly lived in Europe, uh, I don't know, present Europeans will admit to be being descended from Neanderthals. So anyway, uh, were distinct species from modern humans. Okay, or also chimpanzees, as also chimpanzees. Chimpanzees also, we share a lot of genetic this thing with chimpanzees also, our genetic heritage. Okay, maybe 93% of the genes are similar with chimpanzees and gorillas. And of course, they see one thing about humans is we are articulate as compared to our other primates. Okay, now you can't imagine a, I don't know, this is not belittling them. You can't imagine a primate now giving a lecture on this year's Nobel Prizes, right? Huh? They're similar to us in so many ways, but there are some differences, right? Hmm? We are articulate. We can exchange information. We can communicate. Okay, we have so many languages, right? In fact, the origin of languages is again a mystery. Oh, so many languages. We also have 29 national languages, right, in our country. So many dialects. Okay, and how come all this so many languages occurred in so many areas? In fact, the best explanation to me so far is in the Bible. Okay, the Tower of Babel, the Tower of Babel. So the Tower of Babel story goes like this: at one time, everyone spoke one language. Okay, then they decided that they became very quite advanced and all that because they could cooperate and correlate. So they said they will build a high enough tower to reach heaven. Okay, so the Tower of Babel started. Then apparently God got angry. He said, "What are these people thinking? They can come up to my thing." So one day suddenly he created uh, he decided to sort of create discord. So next day they got up, they found they are all speaking different languages. They couldn't understand each other. They fought, they quarreled, and that's all. The tower was stopped, building was stopped. So when I heard the story, I said, "This is ruling by, what do you say? What do they say? What, huh? Divide and rule. Example of divide and rule, right? The British also followed that, right? Divide and rule. And now also is being followed. You divide people, you favor one side, so the other people feel discriminated. You divide and rule. So yes, sure they don't. So this is an example of divide and rule. So origin of language is still a mystery. As I said, origin of our own. Okay. Now of course they think there's a gene responsible for us being more articulate. Apart from the shape of our vulva, the tongue, the back of this thing. It's got a particular vocal cords, they resonate in a particular way, and that's not fully explained. They think that may be genetic. There's something called the FOXP2 gene, FOXP2 gene. Okay, nothing to do with foxes. Foxes are cunning, but maybe we have some of those. So FOXP2 gene is responsible for language, apparently. Okay, so that's what. So it's another mystery which is still not been. What makes us different from other? For example, if you have read Desmond Morris is a famous anthropologist. The Human Zoo, The Naked Ape. So he begins one of his books like this: out of the 193 species of primates, only one dresses and one has rituals for for the dead, and he makes many things. How is it, what makes us different? So again, that's all. When the okay. So these studies then showed that Neanderthals, who mostly lived in Europe, are distinct species from modern humans. So there are some 100 odd genetic differences. However, some of our abilities to resist infection can be probably traced to some of these incorporated DNAs. So our DNA also has a long history, incorporation from other species, okay, from related species. So next, that's all. Huh? Okay, so I try to explain to you although I have not explained in great detail, I try to explain to you why, what is the, how they, what is the reason for giving the Nobel Prizes this year, what is the main impact of that work, okay, and things like that. So yeah, so in fact, the medicine prize could have some important implications for a subject like astrobiology. Suppose you find something on Mars, okay, some organism which were long dead. Now nobody believes that there are live organisms on Mars. They think there was life on Mars once. Okay. Now that's why so many rovers are roaming Mars, right? So many are orbiting Mars. And now the Chinese also have landed their rover there. But the interesting thing is no trace of life has been found so far. Okay. Quite quite odd. Although our planet is thriving with life everywhere, even in the most hostile places. But they are not seen any life. So they think Mars had much water once and all evaporated because of the lower surface gravity and no magnetic field. So solar wind is all bombarding Mars and stripping it of all the atmosphere and the water and all that. So that's why it would have lost its water in a billion years. And atmosphere, escape velocity is there. So the Earth being, higher, much 10 times more massive as a higher escape, 3 times escape velocity. So the probability of molecules escaping the Earth is much smaller. Okay. So on Mars, at any given time, a fraction of the molecules will have substantially escape velocity. So you can calculate. It's a problem in statistical physics given to BSc students how to calculate the time taken for atmosphere to evaporate. Okay. So on Mars, it will evaporate in a billion years. That means long back Mars could have been habitable. They even think there's a new theory which says it had oceans 300 meters deep. Okay, at one time. So then there could have been life at one time. And suppose one of these rovers or some of the future missions finds some kind of life, how to analyze it? So this present method of DNA analysis is important. Okay, we can study the thing and see what drives them. How are they different? It's now believed the DNA is the universal blueprint. Could well be, at least on Earth, it is universal. Any species, right? So in fact, in one of the articles I wrote, right from the Diplodocus. See, Diplodocus is one of the biggest dinosaurs. So 30 meters, right from the Diplodocus to the Diplococcus. See, only the spelling is C and D, right? Diplococcus is such a small organism, you need, even with a microscope is difficult to see. Okay. Right from Diplococcus to Diplodocus, DNA underlies the blueprint of life, the double helix, okay, with the peculiar properties of, unwinding, and breaking up, underlies the, underlies the genetic basis for life. So is it true on other worlds also? So then should be able to analyze, okay. It's still a long way off.